Today I'm going to talk about the two types of precision medicine approaches. The first is based on the multi-omics data. So mostly here I'm going to talk about the statistical approaches. So going through really fast on this first topic because this is AI. The second one is based upon the electronic health records. Here I'm using AI tool to handle the huge amount of data. Alright, so the first topic, a lot of data have been accumulated in the public repository. So here in this study, we generated this for the patient, 80 patient. Which one is the indicator? This one. So in this study, we recruited the 80 early onset gastric cancers. And after we got the tumor tissue and adjacent normal tissue, it's paired. And for each tissue sample, normal or tumor, we generated the exome sequencing data to identify the mutation, somatic mutation, so tumor-specific mutations, and also the RNA sequencing data to identify the genes, mRNAs, upregulated predominantly in the tumor, and also the proteomic analysis. And in this proteomic analysis, we did two types of analysis, the global proteomic profiling to identify the proteins predominantly upregulated in tumor, and also the phosphoproteomics and glycosylated proteomics and glycoproteomics to identify the phosphorylated proteins predominantly upregulated in tumor, and glycosylated proteins are known to be often secreted into the circulation. So we can probably use those molecules as the biomarkers. So here we did glycoproteomics to identify the potential secreted protein predominantly upregulated in tumor. Those are the types of multi-omics that we perform. Using that, it was so fast. Using that individual data, so this is the mRNA data, and we classified the patient into two clusters. So this first cluster was defined by the upregulation of these 919 genes, and the second cluster was defined by upregulation of these 370 genes. And mRNA, according to the mRNA, we can only classify the patients into the two groups. And if you do the same clustering, same patient, using the protein data, this is a protein abundance, so like global proteome data, and you can identify 4 clusters, and similarly, these upregulation of 282 proteins define the second cluster like this. And if you do the clustering, 왜 이렇게 빨리가 이거? Sorry, spoken Korean. Anyway, if you use the phosphorylated level, you can identify 3 groups of patients, and similarly, these 1,192 phosphorylation sites, and their upregulation defines as the first patient cluster here. So, glycosylation. Using the glycosylated proteins, we identify 3 clusters as well. So, we did the individual clustering using individual types of data and to define the proteogenomic. So this is protein data and genomic data. We did integrated clustering. So eventually we identified 4 patient cluster here, subtype 1, subtype 2, subtype 3, and subtype 4. As you can see here, RNA data cannot distinguish this subtype 3 from subtype 4, right? But these additional protein signatures can actually separate them into two. So they can subdivide the RNA 1 cluster into subtype 3 and subtype 4. Similarly, here RNA 2 can be further subdivided into subtype 1 and subtype 2 via these protein signatures. So this is the precision medicine approach. The precision medicine approach normally starts with defining the patient clusters. And then we try to understand the biological or pathophysiological characteristics of each patient cluster. And based upon that understanding, we can stratify what kinds of treatment should be applied to treat the patient in each cluster. So this is the next step. After the clustering, we identify the 4 clusters, and I'm trying to understand what are their characteristics, right? So here, these are the mRNA signature which define the subtype 1, but as I explained in the previous slide, this subtype mRNAs defining subtype 1 could not distinguish the subtype 1 and 2, right? So they are representing the same pathways. But the protein signatures actually, they could distinguish this subtype 1 from subtype 2, so they're supposed to represent different types of pathways. Here, if you can see subtype 1, based upon this mRNA and the protein signatures and the pathways that enriched by these mRNA and protein signatures, we can see subtype 1 has a high proliferation potential, as indicated by the cell cycle related pathways represented by this mRNA and protein signatures. So we named this subtype 1 as a proliferative tumor. And if you look at the pathways that the mRNA and protein signatures, what kind of pathways they represent, then you can see the immune pathways, particularly the anti-tumor killing, tumor killing pathways here. Cytotoxic T cell pathways got activated. That means the tumor killing capability is still present in this type of tumor. So we name them as immunogenic tumor. And if you look at the subtype 3 and the pathways that they're relevantly related, the mRNA and protein signatures represent, then you can find the protein signatures for subtype 3, they strongly represent this metabolic pathway. So we named the subtype 3 as a metabolic tumor. And if you look at these pathways represented by the subtype mRNAs and protein, defining the subtype 4, we can see many invasion or epithelium-mesenchymal transition-related pathways are upregulated and strongly, significantly initiated by these mRNAs and the protein signatures for subtype 4. So we named them as invasive tumor. So proliferative tumor and immunogenic tumor and metabolic tumor and invasive tumor. We understood. So you can expect the prognosis, patient survival, will be a lot better in this immunogenic tumor because these tumors still have the capability to kill the tumor cell by cytotoxic T cell, right? So prognosis is expected to be better. But here, subtype 4, the invasive tumor, right? So high EMT potential, which means aggressive phenotypes. So we can expect that prognosis for the subtype 4 is going to be worse than the subtype 2 at least, right? So if you make a model, they reconstruct a network model to represent this the anti-tumor killing activity. So this is the tumor cell releases tumor antigen, and this antigen presenting cell, macrophages or mostly macrophages. It's very adjacent to the tumor cell. They will phagocytose these tumor antigen and then process them and then present them into the plasma membrane. And this T cell, cytotoxic T cell will recognize the presented antigen and then they get activated and then they shell the tumor cell which produces this tumor antigen presented in this the plasma membrane. So this by see these molecule here, the color in the center, which means the mRNA of this caspase 10 is predominantly upregulated in subtype 2, and the border color, this protein with border color means that protein level was upregulated, predominantly upregulated in subtype 2. And the P and G means phosphorylation of this protein and angular glycosylation of this protein are regulated in subtype 2. By combining all these different layers of the information from mRNAs and proteins and phosphorylated protein and angular glycosylated protein, you can actually draw this picture. If you remove all this information from the protein domain, so this will go away, and this P will go away, G will go away, then this anti-tumor, the immune network will be really sparse. It's hard to see whether the anti-tumor killing activity is still active in the subtype 2. Right? This is kind of power of the proteogenomic approach. By combining all different layers of information, you can better characterize each individual patient group. This is actually what to the clinician, this is not actually interesting because these people live longer, right? So prognosis is okay, but they do care about this patient, the patients in subtype 4 because the aggressive phenotype, right? So if you look at this, Rho A pathways is really popular singer in this area. So it's like a Korean idol, okay? So if you remove all this protein data, right, upstream of and downstream signaling pathway molecule will disappear. So you're not actually going to understand whether the Rho A signaling pathway is actually activated in this subtype 4, right? But by combining all different domains of data, then you can better understand, and we can see clearly now. Both upstream factors and downstream factors are activated in subtype 4, which can induce that aggressive phenotype shown in this subtype 4. So, based upon this understanding, so if I can go back a little bit here, subtype 4, they have high EMT, invasion potential, aggressive, but they don't have any anti-tumor immunity here, which means immune cold. This tumor subtype 2, immune hot, right? Immune system is suppressed and the invasive potential is so high. So what kind of treatment can you propose? This is the multi-omics based precision medicine approach, which normally goes by group, patient group. It cannot be personalized. So, because the only thing you have now is the understanding of the group characteristics, right? Based upon that understanding, you can propose only the therapeutic options for each individual group. So, this is the important, or the more clinicians mostly care about, right? Subtype 4, because this is aggressive. So, it suggests that you need to inhibit this invasion, increase the invasion potential. You need to lower that, right? So you have to inhibit this Rho pathway, Rho signaling pathway. And by looking at why these tumors, these kinds of tumors became immune cold, and you can suggest many molecules, right? The myeloid-derived suppressive cells, this is the innate immunity level. Inhibitors of the T cell activation, cytotoxic T cell activation, and immune checkpoints. And this MDSC mostly we found they are the PMN, this is the mononuclear, the polynuclear, or the neutrophil mostly, and multinuclear, right? This is the neutrophil mostly. Neutrophils got dysregulated, so you expect to see many neutrophil extracellular traps present in these kinds of tumors. And also immune checkpoints get elevated. For example, like the PVR was highly expressed. So you can probably think of PVR inhibitors, so which TIGIT normally. So you can combine all this. So this group level suggestion is combine the therapy to inhibit this raw A signaling pathway, and also you have to inhibit this MDSC by by subscribing, administrating the IL-10, something like that, and also the TIGIT together. So this is the multi-omics-based precision medicine approach. How much time I have? 몇 시야? 얼마나 했어? I don't know how much time I have, but let me go. I was thinking actually go really fast for the first topic, but anyway. So this is the deep learning approach here. For type 2 data, so for the first topic was about the gastric cancer. I applied the same approach for pancreatic cancer and the cervical cancer and prostate cancer as well. And now I'm shifting into the AI modeling here. I'm mostly applying this to type 2 diabetes and the dementia and what else I have. And many diseases, several diseases. So, from the Seoul National University Hospital, we have two major branches here. This is the headquarters here, and this is the main hospital, and this is the second branch hospital in Gundam. And for each patient, they regularly visit this institution, right? Every 3 months, basically in Korea. And every time they visit, they get the test, blood test and the urine test. So mostly every time they visit, we can gather like 130 variable, and the measurement for those 130. So I listed some of them, sorry for Korean. But here we carefully reviewed because we used the patient who visited these two institutions from 2004. At the time, the measurement, regular visit was there because they have to get the prescription for the drugs they have to take. But the number of variables, at the time was like 54, 60. So we wanted to have a more complete data set. And those are the variables kind of traditionally thought to be related to this type 2 diabetes. So we used this 60, about 60, 59, and the medication as well. So we did this 3-step modeling here. Initially, we treated the measurement at different time points for the same patient separately in this stage here. So we are summarizing, we are embedding into the low dimensional space using variational autoencoder. But here we treated the different time point data from the same patient separately, just like independent observation. And then here in pre-training, the embedded in the low dimensional space, so we normally call Z values, they used as an input to this transformer here. So the embedded value will be used as input values for this transformer. But here we now combine, because we have to define the positional encoding. So we combine all the measurements measured at the different time point for the same patient as one data. And the temporal transition should be understood as a positional encoding, so sequence. So Here, from the same patient, multiple measurements are treated separately. Here, all the measurements from the same patient will be treated as one observation using positional encoding. And then this is pre-training data. We learn everything from whatever patient which have this 59 variables. But for fine-tuning, we wanted to do the transfer learning to the pre-trained information to predict HbA1c level and the EGFR level, glomerular filtration rate. So during this fine-tuning, we only use the patient who had the HbA1c measurement at the time. So here, we don't care about whether the patient had this HbA1c measurement or not. But here, we're only using the patient who had the measurement for HbA1c. This is schematic, and you guys will know better than I do, right? So this is pre-training, and if you look at the attention, and this attention map kind of indicate that the neighboring, the time point information will mostly affect or contribute to explain the sense time point. So here, I'm standing here, and if I look at the different time point information, this neighboring information will be more important, right, to predict the measurement level at this time point. Right? That's what this pre-training, the attention map shows you. And if you look at the individual variable here, and if you do the decoding using the transformer, and you can see the embedding is really okay. And we developed the separate transformer pre-training, the fine-tuning model for clinical data. This is mostly continuous data. And this is the drug administration data. We had about 20 different drugs. And if a particular patient at this time point used the drug A, then we did one hot encoding, like the data representation for this drug model. So all this is variation of sorry. This is the embedded data in the low dimensional space from the pre-training transformer, and then we use that transformer to do the decoding using this decoder, right? So if you look at here, the model kind of okay to predict this HbA1c model. If you use the clinical information only, the R-square value is 0.89, and if you use the drug information only. So in this model, I'm not using this clinical data, I'm using the only types of drug administered at the time. And if you look at using the drug information only, you can see the R-square value at 0.8. So it drops a little bit, which means the doctors do not understand correctly then this AI model, right? This is the clinical information. By looking at those clinical data, they are choosing what kinds of drug they should administrate for the patient. But anyway, so that's the reason why the R-square value change differs between these two models. And if you look at the attention map now trying to find the variable temporal correlation, right? So you have to start to see the off-diagonal values highly kind of increased compared to the pre-training. Pre-training only cares about time, right? So neighboring time point should be treated importantly. But to explain this HbA1c, you need to look for other correlations among the variables like the calcium level, cholesterol level, right? So you start to see the offline, the attentions, and if you do the evaluation study, you can quantify the importance of each individual variable. So the fasting glucose level and glucose level obviously should be more important, and the liver damage indicators, ALT, ALP, they are within the top 10. And we did the drugs. Drugs, interestingly, let's skip it. Anyway, important drugs found actually. So we did this is the UMAP, which shows the distribution of each individual patient in the latent space, right? And if you look at them, you can cluster them. So there's a cluster 2, and if you look at their cluster, for each cluster, if you look at the actual level of this HbA1c, you can see these patients, even if you make any drug, if they don't respond, actually, HbA1c does not go down. And if you look at these patients, they easily go down with the medication. And here, they go down, but they will stop at a certain level, right? And even if you are treating many different drugs, these patients, HbA1c still keep going up. So these are the kind of troublesome clusters, the patient groups here, these two. So if you look at what kinds of variables are contributing to defining this patient group, you can see the ALT and AST levels are high, which means these are the patients with the liver damage. So if you see the patient with the liver damage, and you should treat the liver damage first, and then do the diabetes care, right? And this is the region. And if you look at these kinds of patients, some patients with the drug administration, the HbA1c will go down a little bit. In the beginning, it's okay, but it doesn't go down all the way down below this threshold here. So these patients, if you look at them, you see this urine creatinine ratio is high, which means kidney problem. So if you look at the patient which shows this kind of pattern, you should care the kidney together with the diabetes. That's what that means. And then this is the cluster level understanding. So it is very similar up to this level. It is very similar to the multi-omics approach, right? Multi-omics group level understanding and then do the treatment, right? You should do the kidney treatment and the diabetes together. But here, since you have a model, you can do now personalized the precision medicine here. So this is the once this particular patient visited and you have this clinical data, and then you don't have this drug because you are the doctor now. By looking at this clinical data history now, you can propose all kinds of drug. And then we let the AI model do that task here by just enumerating all prescriptions that the doctors prescribed for past 18 years, right? And then we chose the best drug which lowered more effectively this HbA1c. So model understands that this is the real drugs that prescribed by doctors, and this is the drug selected by AI model. And this is the real profile of the HbA1c of this patient based upon this drug treatment here. But so the model understand it will go up. So they are suggesting stronger medication here. This the early stage here. This is the metformin and this is the DPP-4 inhibitor. So stronger medication first, and then once it's stabilized, you can combine with the metformin. And this patient, this is a severe case. This is a light case, and this is a severe case. If you do the same thing, the model understand it will go up. So it suggests kind of a more stronger medication in the beginning. And this case, this is a pain in the neck, and because the doctors don't understand how these patients will move, because when they visited first time, the institution, the level of the HbA1c is almost 9, close to 9, which means really serious clinical condition here. So doctors don't know how these kind, because they haven't seen these kind of patients many times. They don't understand. They don't have any knowledge on these patients. And the model also couldn't learn the proper knowledge for these kinds of patients. So both fail actually. So we need to collect more data for this kind of patient to improve this AI model we have. Okay. All right. Thanks for listening, and I thank everybody.